Okay, I'm now going to lecture to the transcription PowerPoint. And if you look at this first slide, it's just, we had the slide on the previous PowerPoint. It's just an overview of the central dogma of molecular genetics and the idea that the DNA stays in the nucleus, that there's transcription of the DNA to create the mRNA molecule, which then leaves the nucleus and is acted on by ribosomes to create the protein. So for this PowerPoint, we're going to focus in on the process of transcription and how that works. So before we get into the process, I just want to use this chart to show you the different types of RNA. We've only mentioned one so far, and that's the messenger RNA, which is made in transcription, and it's what gets translated into protein. We will see these two here, these types of RNA, when we talk about translation. And we'll briefly mention this small nuclear RNA a little bit later in this PowerPoint. You're not going to be responsible to know these other types of RNA, but I just want you to be aware that there are multiple types of RNA. So obviously in transcription, our focus is the mRNA, which we talked a little bit about before in the previous PowerPoint. You should know where it's made. So that's the nucleus. How is it made? Well, it's complementary to the DNA sequences in the genes. And if I can use the analogy of the DNA molecule in the nucleus as a recipe book, then the mRNA is just a one single copy of a recipe from that book. So if there's maybe thousands of recipes in the book, the mRNA is specific to a gene. So it's one recipe from the book. You're making multiple copies of it, and that's being shipped out into the cytoplasm to be acted on by the ribosome. So to create the, meal from the recipe, the action of that happens in the cytoplasm. So each mRNA is copied from one of the two strands of DNA using the same process as what we learned in DNA replication. So there's a complementary base pairing that happens, the same sort of thing that happens in DNA replication. So as we start to move through the different steps of transcription, I want you to think about a comparison between transcription and transcription and DNA replication. They are similar processes, but there are differences. So perhaps when you're studying, you create a table to look at those similarities and differences between the two. Each of the mRNA has a 5' and a 3' end, with each ribonucleotide added to the 3' hydroxyl group of the growing chain. So that is very similar to what happened in DNA replication when the DNA polymerase 3 is adding the bases. That was also to the 3' hydroxyl end. So instead of using DNA polymerases, transcription uses RNA polymerases, which adds ribonucleotides, not deoxyribonucleotides. And it always moves in a 5' to 3' direction, which is the same as what was happening in DNA replication. So this whole process of creating the mRNA is called transcription. So there are three phases. There's the initiation, the elongation, and the termination. So we'll first look at the details of initiation. So the RNA polymerase binds to the DNA at a specific region. It's called the promoter region. And that region is found upstream from the beginning of the gene that's going to be transcribed. And you'll see in an image coming up what I mean by upstream. This region where the RNA polymerase binds is very high in A's and T's. It doesn't mean it only has adenine and thymines. It has other bases as well. It has the G's and the C's as well. But it has a lot of A's and T's. And these are called TAT boxes. And it is believed that the reason why the promoter region contains these bases is because the number of hydrogen bonds between A's and T's is only 2, compared to 3 between G's and C's. So it takes less energy to break those hydrogen bonds. And really what's happening when the RNA polymerase binds to the promoter region is it's opening up, it's unwinding the DNA. So there has to be the breaking of those hydrogen bonds. So having those A's and T's in that area makes it easier for the RNA polymerase to do that. So here you can see the stretch of DNA that's going to be transcribed. So this is the gene, also called the transcription unit. And you can see the RNA polymerase here the enzyme is binding to a region that is further upstream from the DNA. It's binding here and then will move this way across the transcription unit to create the new mRNA molecule. So now we reach elongation. So this is the actual building, the adding of the bases. So just like what's going on in DNA replication, but it's only happening on one strand. So there's no lagging strand or leading strand. So it's going to be reading the template strand of DNA. It's reading it in the direction of 3' to 5' because it's creating its building in the 5' to 3' direction. So it can only work in that direction, just like we had with replication. What's different about transcription and DNA replication is there's no primer required. And it's important to note that that promoter region is not transcribed. That promoter region never turns into protein because it never turns into mRNA. It's just used as a spot for the RNA polymerase to bind to. So the strand that's being read is called the template strand. Well, it's also called the antisense strand. The strand that's not being used as a template, so the other strand, is often called the sense strand or the coding strand. So once the DNA is transcribed, so once the mRNA is produced, the DNA gets rewound back up and the polymerase moves off of it. So we're going to start to see some diagrams. This diagram shows the promoter region. So this area here is considered the promoter region where the RNA polymerase will bind. And you can see that it has a lot of A's and T's in it compared to G's and C's. So those are considered TAT boxes. So the RNA polymerase will bind here. Notice that there's two strands indicated here. There's this strand, which is acting as the template strand, also called the antisense strand. And then this other strand is the sense strand, or the coding strand. Okay, so once you read this little green light is supposed to indicate where transcription actually starts. So this part here is not getting turned into mRNA. Only when it hits this spot here does the transcription begin. And you can see that this is the template strand. So we're going complementary to the template strand to create the mRNA. But notice that instead of a T here, we're putting a U because it is RNA. So uracil instead of thymine. So we're building it into 5' to 3' direction, but the template strand is being read in the 3' to 5' direction. Now, one of the reasons why this other strand, the sense strand, is also called the coding strand is if you look at it up here, it really matches up with what's being built with the mRNA. The only difference being there's a U instead of a T. So that's why this is called the coding strand. Okay, moving on. Here's another image. This is the RNA polymerase molecule, very big enzyme. This is the template strand. The red here is the building of the new mRNA molecule. So once a sequence is reached called the terminator sequence, the RNA polymerase knows it's done and it's going to start to dismantle or disassociate from the DNA. Now, we're not going to get really into the details of what this sequence is, but you should know that it's different in prokaryotes and eukaryotes. And as we move through this section of the unit, really try to make note of the differences between prokaryotic genetics and eukaryotic genetics. So as I said, the mRNA will dissociate from the template strand, and that would stop the transcription. And now the RNA polymerase is free to bind with other promoter regions. So that's the termination phase. Now, you can see a series of diagrams here. You can see that the RNA polymerase is binding to the promoter region. It starts to move along the transcription unit. This little red thing is at the beginning of the mRNA molecule that's being created. Now it's moved quite far along. So a lot of it is produced, and then when it reaches termination, everything dissociates, and there's your completed mRNA transcript. But it should also be noted that as this one RNA polymerase is moving along the transcription unit, the promoter region is free to have another RNA polymerase bind to it. So you could have an RNA polymerase here and another one is binding here. So you're making many copies of the mRNA. It's not like you just make one copy and that's shipped out into the cytoplasm. Obviously, the cell needs to produce a lot of the protein, hundreds